Hola, bon dia de nou. Ara passem a fer la conferència. Se sent? Gràcies. Passarem a fer... Tampoc? Passarem a fer la conferència de clausura que presentarà el conferenciant, el Kostas Kostarelos, el presentarà el Xavier Navarro, però abans vull agrair molt al degà de Biociències, a l'Isidre Gibert, que estigui aquí per fer la clausura de totes les jornades. Les biojornades dels 4 departaments de Biociències van començar el dimarts i acaben avui amb el BCFI. No acaba la jornada amb això. Per la tarda tenim conferències i xerrades plenàries i xerrades molt interessants fins a les 5 de la tarda i premis a partir de les 5, però l'Isidre no ha d'esperar-se fins a les 5 de la tarda per venir a donar-nos l'acte institucional de cloenda de les jornades.
Molt bé, gràcies. Sí, se sent? Sí, Sí, no ens en calen dos, Res, simplement agrair-vos, en primer lloc al departament per haver-me convidat per fer aquest acte de clausura d'aquestes biojornades. Per mi és un goig poder estar representant en la facultat en aquest moment, també en els que he participat en els altres departaments, però especialment en aquest perquè estem tancant un moment molt interessant per tots nosaltres, i dic interessant perquè el que veiem o el que hem vist i el que veurem encara, la tarda encara seguim, en aquestes jornades és un clar exemple del que és una facultat universitària, on no sols està present la docència, que això és evident, sinó que molt important està present la recerca que fem, que feu totes i tots, i que ens permeten traslladar aquest coneixement a la nostra docència. I això dona aquest valor afegit, aquesta qualitat afegida en els nostres ensenyaments. És un moment, i ens ha passat, estem en la 7a edició, 8a edició d'aquestes biojornades, i el que hem anat veient i el que hem vist també aquests 3 dies i avui, que és un moment per compartir, per conèixer-los, sobretot en departaments com el vostre, en el que hi ha, ara comentàvem, 4 àrees de coneixement, 3 àrees de coneixement, però que esteu molt dispersos en el campus i fora del campus, i és un moment en el que podeu compartir i conèixer els que teniu el mateix replà on viviu, perquè això ens passa tot sovint. Som veïns però no sabem qui és que viu o què és el que fa el que tenim al costat. Poca cosa més, que és un moment això de compartir, de conviure i de donar-se'n de la qualitat de tot allò que fem al voltant d'aquestes parets que ens aixopluguen. Gràcies de nou per la vostra assistència, gràcies al departament per fer-me partícip d'aquest moment, que és solemne, perquè estem acabant, i poc més. Gràcies a tots.
Gràcies, Xavier, sisplau.
Bé, en primer lloc, agrair a la direcció del departament i al comitè organitzador que haguessin acceptat la proposta de conferenciant que vaig emetre i per permetre-m'hi estar aquí amb vosaltres presentant-ho. Faré la presentació en anglès perquè Kostas pugui copsar si dic alguna impertinència respecte als seus mèrits. So, Kostas Kostarelos, as obvious from the name, from Greek origin, but he made his graduation and doctorate in chemical engineering in London at the Imperial College of London, one of the best universities in the world, as by the last rankings. And there he studied liposomes. Then he moved to the USA to make several postdoctoral training periods in excellent institutions, like the Memorial Sloan Kettering, University of California, San Francisco, Cornell. Then he came back to London in 2002, about 20 years ago, and joined the Center for Drug Delivery Research at the UCL School of Medicine. he became head of the center, and then by 2013, 10 years later, he moved all his lab, large lab, to the University of Manchester, where he became leading the biomedical technologies area, and he's now chair of nanomedicine at the Faculty of Biology, Medicine and Health, and also director of the National Graphene Institute. In 2016, he became the leader, director of the Biomedical Technology section of the Graphene flagship. As you may know, one of the two main research adventures that the European community make, one for brain and the other for graphene. And he has been director of that area until now. By 2020, Kostas incorporated part-time, he's about 20-25%, at the ICN2, the Catalan Institute of Nanoscience and Nanotechnology, that some of you may know, that it has a building here in campus, where he has started a new research group on nanomedicines. Okay? So he's sharing part-time here in Bellaterra and part-time in Manchester. He is very interested in opening cooperations, particularly of his great knowledge and great capacities in developing nanomedicines and nanostructures with biomedical and biological groups. And that's one of the reasons that we proposed him to give this talk. Now, my group, myself, we are already cooperating something with Kostas, but he is very open to establish more cooperations with any of you, and that was one of the main reasons that he wanted to present the talk today. He has been telling me that in the last 10 days he has like 4 talks in 4 different countries, so it's a little bit stressing for everyone. But he is delighted to share with all of you the events. Any of you who wants later on establish contact, he may be around here until the afternoon, or eventually by email. He has an email at the ICN2. You can contact him and establish any potential meetings for future cooperations. So nothing else, Kostas. Yes. The stand is yours. Thank you very much for coming.
Okay. Hello. Can you hear me well? Yes? Okay. Good afternoon, good lunchtime. I don't know what you say in those things. I'm very happy to give the closure to talk of your away day or department day. The reason why I'm particularly interested giving this talk, as Xavier mentioned, is to try to tell you and give you a kind of flavor of how you may start thinking of nanoscience and nanotechnology that I am assuming you do not hear very often about. I'm going to give you 2 stories today. I brought 2 stories with me. I've given a talk at the Neuroscience Institute, in case some of you were present, about 3 weeks ago. I will not present anything from that talk. So I promised Xavier that I'm not going to show anything on that talk. So I'm going to give you 2 stories, which are 2 very different technologies. The only similarity between what I presented earlier at the Neuroscience Institute, which was very device-oriented, and here is only the material, as you will see of the second story. The first story is not going to be at all relevant to what you've heard before, but it's a very new technology that we have developed. So let me start by... Oops. Let me start, if this plays. Let me start by showing you these 2 schematics that I'm beginning our lectures with, which are really demonstrating the array of different nanoparticles and nanosystems that one could possibly have access to and design different technologies around. I call this one the older toolbox. This is because most of these nanoparticles that you see here on the left-hand side are invented 20, 30 years ago. And the other reason is because on the very right-hand side, you see that all of them have a clinical presence. I'm only going to highlight a few for the interest of time. Liposomes, which is going to be the main nano system that I'm going to feature in the first story I'm going to tell you today. And the other ones are viral nanoparticles that I'm sure a lot of you are familiar with as a biotech, micro technology department, Institute Cell Biology. So these are systems that are falling under the nanoscale umbrella. So maybe you think that they are just delivering genes for you, but if you look at them from a structural point of view, they are nanoparticles. In fact, they are beautiful nanoparticles because they not only are very reproducible structurally, but they're also extremely smart and efficient in delivering their genetic cargo and information. I will not talk about viruses today. Then we have a genre of nanoparticles at the top, which are metallic nanoparticles. Some of you may have worked with them or have seen papers. Silver, very famous, very well-used antimicrobial agent. A lot of domestic products contain silver nanoparticles today. And another example is iron oxide nanoparticles, in case you're involved with magnetic resonance imaging. And these are solid, spherical, most of the time, nanoparticles that are used, again, in many different applications. The last one is only an example of a polymer system. It's called Dendrimer, but this is just a representation of a huge field of polymer science. If there's any chemist in the audience, will perhaps be offended by me just putting dendrimers here. There are hundreds, maybe thousands of iterations of polymer nanoparticle systems that are used for biomedical applications. Again, all of these are the result of work that has happened in the last 30, 40 years. They are not really novel, but their applications could be novel. So the first story I'm going to tell you is about liposomes, which are lipid bilayer shaped spherical nanoparticles. They happen to be the savior technology from COVID as well, but this is just a coincidence because, as you probably have read, the Pfizer and the Moderna technologies are both based on the same structure, which is a lipid nanoparticle which contains the mRNA components. I will give you a completely different use of how one could fabricate and engineer a liposome or use a liposome for biomarker discovery studies. The second toolbox is what I call a younger toolbox. I call it this way because it consists of nanoparticles, nanosystems that have been discovered much more recently. And the second reason I'm calling it younger is because it's clinically more immature. So there's, as you can see for the far right column on the slide here, there's maybe not any clinical evidence of how these systems will behave in a human body, and there is only some preclinical evidence of how they would perform once injected in different routes of administration in a human body or even a large animal species. I'm going to focus, because of what Xavier mentioned, I have been kind of one of the kind of fighters to put graphene and flat 2D materials in the biomedical space for the last 10 years or more. So I'll give you the second story I'm going to tell you is how to design graphene nanoscale sheets of 2 dimensions as transport systems for therapeutic development. So let's start with my first story. The liposome system, as I mentioned, is a system that was discovered or kind of developed in the mid-60s. The first liposome reference is 1965. And it has been mainly used as a vehicle for the delivery of different cargoes of biological activity. I'm going to tell you one thing, and I think everybody in the audience will say, it's a liposome, lipofectamine. Okay? All the gene transfer agents you've used were liposomes, are liposomes. I'm going to give you a completely different twist now in how you can use this technology that we came up with about 5-6 years ago. The whole concept, technologically and from a scientific principle point of view, is based on this phenomenon, which is called the corona formation once you place any nanoparticle into a biological fluid. As you probably know, a biological fluid consists of a very rich collection of biomolecules. These biomolecules will spontaneously adhere onto the surface of the nanoparticles once they are placed into a biofluid. So even if you have a nanoparticle that has a polymer surface property, if it's positively charged, negatively charged, if it has other proteins on the surface, there's always going to be some degree of adsorption from the medium onto the surface of the nanoparticle. That's called, in the nano space, traditionally the protein corona. So I'm going to tell you the beginning of the story with this paper here, whereby I had a PhD student who was telling me, well, I'm not sure what I want to do. I want to do Alzheimer's and blah, blah, and I want to use liposomes to deliver an Alzheimer's drug. So I told her, no, we're not going to deliver stuff anymore. Let's do this. Let's try to get liposomes into an animal and then recover those nanoparticles from the blood circulation, and then examine all of the biomolecules that have coated those liposomes and see what we get. And she hated me for this. So this is the result of this conversation that lasted about 2 years. So we injected liposomes, empty, these are empty liposomes. The liposomes were used from the very beginning are the clinically approved liposomes. I mean, I'm not sure if you're familiar. In 1995, the FDA approved the first liposomal drug, which is a liposomal doxorubicin called Doxil or Calyx, depending on which country you buy it from. And this is a pegylated, a polymer-coated liposome that contains doxorubicin, as I will show you in the next few slides, in its inner phase. So a typical structure of a liposome under cryo-electron microscopy looks like this, perfectly spherical shape. The lipid bilayer here, aqueous space in the middle. You can fill it up with drugs or mRNA or whatever you want to deliver, and then you can play with the surface properties. So we took those guys, not any drug contained in this particular case. We can make them in our laboratory very, very reproducibly, very consistently. We injected them intravenously. Then we waited between 10 minutes and 1 hour. Why did we wait? And this is the advantage of working with an old nanoparticle. We knew the pharmacokinetic profile of those liposomes very well. So we knew that the blood will be full of liposomes within the 10-minute to 1 hour time frame. So we extracted all of the blood. We had to sacrifice the animals by cardiac puncture. We took all of these. We separated the liposomes from the rest of the blood components, and then we purified them. I don't have time to go into, plus you're biologists, so you don't care about all of these boring purification techniques that chemists do. And then we looked at the nanoparticles as they were extracted and purified. So this is how they look like under cryo-EM after this purification step. And you see immediately that the surface of those particles is already kind of noisier. There's got stuff on it. So then we did a step forward, and we said, okay, we got what we wanted. We have a protocol now. We can extract liposomes from intravenous injection. We let them interact with the biomolecules in the bloodstream. Let's go do it in humans. And that's the advantage of using, again, an old nanoparticle. So we collaborated with the cancer hospital in Manchester, and we knew that there were ovarian carcinoma patients at late stage that were administered with liposomal doxorubicin. So this is the product here, Doxil, as I mentioned. So we put together a very simple clinical study whereby we were there after the infusion, after these patients were infused with the liposomes, and all we did is at the end of the infusion, we picked up blood. And we knew that this blood is going to contain liposomes. We took these liposomes and then we followed the protocol I showed you in the previous slide. And you can see even from the STS page how technologically a step up is in terms of protein, at least, fidelity. This is the plasma of the patient, of one patient, if you do not extract the liposomes, if you just take plasma alone without any liposomal extraction. And you see the problem here is called the albumin masking problem. All of the large molecular weight, highly abundant proteins will smear your gels. And you will lose fidelity and detail of what molecules have absorbed on the surface of the nanoparticle. If you do the nanoparticle here, protocol, you'll see that you start getting a very clear revelation of all of the different molecules that have absorbed on the surface of these objects. Now, if you run mass spec following that, you'll find an extraordinary increase in the number of proteins you can identify. And again, in these studies I'm going to show you here, we're only looking at proteins. I'll show you at the end of the story how other biomolecules are behaving. So we were kind of convinced that in both mice and humans, the nanoparticle corona can enrich the proteomic information that we can extract. So that's why we were calling them nano scavengers. So we thought that the liposome is acting as a template which allows proteins and other biomolecules to absorb, and then if we pull it out from the biofluid and analyze it, we have a much richer and much more high definition picture of all of these molecules that absorb on the surface. However, how can you do this in clinical practice? You cannot do it. I cannot start injecting you with empty liposomes. I don't think any of you would agree to that, even if you were paid quite a lot of money. So what we did is we switched for all of our clinical studies, and we looked at this same technology, but instead of injecting the patient, taking the blood out, take a blood sample from the patient and immersing the nanoparticles into the blood sample, what we call the ex vivo corona. And that has opened up a whole range of different, by following this pipeline that this slide is showing, is opening up all kinds of possibilities in terms of clinical usage of this technology to discover unknown potential biomarkers. How does it work? You take the blood sample from the patient at different stages, before treatment, after treatment, et cetera, so it depends how you design your cohort with your clinicians. You are immersing the blood sample, the nanoparticles into the blood sample, extract, purify, exactly the same procedure I showed you before, and then you run mass spec, and you have a clear differential between the healthy cohorts and the diseased cohorts. So in order to run a biomarker discovery study, you need to have the healthy equivalent, age-matched, sex-matched equivalence. Or if it is a particular study, I would suggest you start, the best studies for me are the ones where you have the individual and you longitudinally monitor them, but this is a very challenging from a practical and clinical point of view how to collect those samples. Anyway, in this particular case, we had groups of healthy and diseased, and I'll show you one example how we use this technology to discover new biomarkers in ovarian carcinoma. And then, once you've identified which of these molecules are highly differential between the healthy and the diseased, then you can make your picks. And depending on different analysis and analytical tools that I don't have time, and I think if there are any bioinformaticians in the room, they will know much better than me what to do at that point. You select your potential biomarkers, which you then validate by following ELISA. So you go back to the original sample without any nanoparticle. You run ELISA on particular targets. But at this, you use this exercise for target identification purposes. So I'll show you an example how we've done this and how we've illustrated and planned this. This is case study number 1, ovarian carcinoma. So we had 20 ovarian carcinoma patients, late stage, again, similar. We took blood samples before any injection of liposomes. We immerse the liposomes after we've extracted the blood. And then we had age and sex healthy controls, matched healthy controls, 10 of these. And this is how the proteomic information analysis looks like, and you see immediately that you clearly have groups of proteins here that are completely absent from the healthy cohort. Just to give you one example. So immediately you see that, oh, there's something going on here. If it was similar across the board, you will be seeing something like that. So then you would need to do a much deeper bioinformatic exercise in order to decipher which ones. You may still have, by the way, interesting targets here, but you need to dig deeper a little bit bioinformatically speaking. So what is the step? The next step? The next step is to run the mass spec analysis and see which ones are either downregulated or upregulated, which of these molecules are. So we found that in this particular case, 413 differentially abundant proteins, either up or downregulated, but they were differentially different between the healthy and the carcinoma cohort. And then we've identified 9 of those targets for the reasons I'm going to show you in the next slide. So we've identified 9. These, we had one group, which is the orange. So targets, these are protein targets, which we're going to run ELISAs with. And the green, three green ones, and 3 indicated as this purple-blue color. And the reason we wanted to do that is because we wanted to run the validation with clinically used biomarkers. So we knew that in ovarian carcinoma there are clinically used biomarkers. The most famous one, perhaps you've read about, is CA125. It's not a very good biomarker, by the way. There are papers that are completely thrashing its usage, and we wanted to see how it will behave. This particular target is going to behave in the context of this technology. So these 3 are clinically used biomarkers in ovarian carcinoma. And you see that with this analysis and this validation with the ELISA that we perform, the sensitivity to specificity curves that we find are giving us numbers around the 0.9 range. The perfect one will be 1, by the way. So you want the perfect sensitivity to specificity. The perfect target would be the perfect, extremely sensitive, extremely specific. The second group were proteins which we had identified, 3 proteins we had identified, which were previously associated with ovarian carcinoma pathways, but were not clinically used. And we saw that with these 3 molecules, the results were becoming more interesting. And you see how this expresses in the sensitivity specificity curves. They are closer to 1 now. And then even more interestingly, we decided to pick up from our mass spec molecules that are on the edge of differential abundance. So the ones that are dramatically different between the healthy and the diseased samples. And you see here that we're getting even some molecules that are spot on 1, so very spectacular sensitivity, specificity. And with this exercise, I just want to illustrate close the example. It's published in this journal, Nano Today, it's called. And you can see that the protein corona analysis of the nanoparticles allowed the discovery of previously unknown potential biomarker protein panels. Of course, the difficulty here, and I will leave that for discussion if you wish, is more of how do you clinically translate it? How do you take it to the next level? Because these are 20 patients. We, in an academic lab, we don't have the capacity to do this validation exercise with 2,000 patients. And this is what the big problem, I mean, I've been discussing with a lot of people also a couple of weeks ago here in Barcelona about how do you go about if you do not have a company that's going to put out the cash and the resource and the capability and the sample availability from the clinic in order to be able to identify and validate those markers forward. But I guess this is where the limit of academia is kind of reached. But let's talk about technology a little bit more. I'll give you a second case study of how we use this technology. And I think some of you would like, I saw already some Alzheimer's models by some of you out in the posters. We took this double transgenic animal of Alzheimer's disease, the APPPS1, and we compared, and wild types. So we have those 2 cohorts of animals, the double transgenic and the wild type mice. Now, for those of you that are in this Alzheimer's field, you know that this particular model is very, very specific because it gives you within the time frame of 1 year this kind of pathophysiology, which is in 2 months you see very few plaques, and then in 12 months you see it's full of plaques, plus you start having severe behavioral memory loss. So it's a very clear model in terms of usage. And this is the advantage of using this technology, because now we can do it in vivo. Because now preclinically we can inject those nanoparticles, recover them from the bloodstream, get the maximum fidelity of the analysis that we could get, and map it longitudinally to disease progression. And this is a huge challenge, because we are trying, as I speak, gathering samples from across the world from patients to run similarity in the longitudinal cohorting blood samples, and it's becoming very, very difficult for a variety of reasons I don't have time to go into. So if you look at the mass spectrometry data of abundance, in 2 months you see groups of upregulated proteins and some downregulated proteins. Again, I'm only showing you proteins that are very different between the wild type and the Alzheimer's animals. Very interestingly, in 12 months, you see more or less the same proteins that were upregulated 2 months. In 12 months, they are downregulated. And that is very interesting and very important. I think that's what the study has kind of, first of all, identified, that perhaps the lack and the difficulty in identifying credible biomarkers in Alzheimer's is because there is a dynamic nature in which the molecule is expressed and circulating in the bloodstream of patients or those animals, depending on disease progression or the overall physiology of the patient. So you see very clearly here that 2 months is giving you most of the proteins, the differentially abundant proteins, at the upregulated stage of the map, and the fold change, upregulation fold change, whereas pretty much the same molecules at 12 months are downregulated. So then what are you designing for? Which market do you pick? And the last point I want to make is that you can also draw bioinformatics analysis and molecular pathway analysis with those results, because again, you want to map the molecular pathways that you see upregulated involved, either as upregulated or downregulated, again mapping it to disease progression. And again, this is something that if we were running the same study from plasma extractions, we wouldn't be able to do it, because albumin will be in the way. So that's why I want to tell you, and I'm going to leave you with, that this technology, we think, has allowed the discovery of longitudinal blood proteomic fingerprints. Now, again, my plea, if you have models, if you would like to study longitudinally disease progression, this is a technology that perhaps can give you a much richer, at least at the proteomic level, data set in which you can then dig deeper and cross-check your molecular pathways and the target proteins that you think may be involved in the different diseases you're studying, but in a longitudinal fashion, at least at the preclinical stage. So just to summarize a little bit what this technology and what I've showed you that we can do the same, we can follow and perform the same principle in 3 different types of samples: plasma samples, mouse models, and humans. You put the liposome in the biofluid, take it out, purify it, and then you have this protein corona that is very rich, very high fidelity. You open up and you solve 3 problems in proteomics. 1, the signal-to-noise ratio is highly improved, as you can see here, comparing plasma analysis to the corona analysis. The second is that you open up the dynamic range of molecules, so the number of molecules you can identify is much, much larger because there's no masking anymore. And the third, and very important, at least in cancer, I'm sure in other diseases, is that you are discovering molecules that are in this area of the spectrum. This is molecular weight, and this is abundance, amount of protein in the blood. We know that most of the interesting biomarkers will be of small molecular weight and will not be as highly abundant. They will lie here. They will be small molecular weight and low abundance, whereas the proteins such as albumin will be kind of up here, high abundant, high molecular weight, which will smear. We have a package now, a bundle of IP that we are trying to license different companies. And again, I'm going to reiterate the difficulty in doing this. And as an academic, I'm also reaching my kind of limit with regards to how much negotiation power and skill do you have with the different companies that are trying to use this type of toolkits. Okay, I'm going to close this example and this story with one more futuristic kind of, where does the technology go forward? How are we taking it forward technologically speaking? The step up that we've done and we've published last year is in this what I call nanogenomic scavenging. As you probably know, at least in cancer, neurodegenerative diseases not so much, but in cancer where biomarkers are kind of dominating the scene, genomics are proving to be extremely popular and very, very useful. So what we set out to do is ask basically the question, if we follow the same protocol, do we get any cell-free DNA on the nanoparticle corona? And the answer, cutting a long story short, is yes. Why? Because by this graph, you can see very clearly that the liposomes are pushing the detection of cell-free DNA, which would be in these blue bars, and this is the fractionation. This is the chromatographic fractionation of the sample that we have from the liposomes. You see here, if you have no liposome, here, the blue, you get the DNA coming out in multiple fractions. If you do this liposome trick, where you put the nanoparticle and take it out, purify, and check for DNA, you have these red bars, which means you're pushing at a particular fraction, which, by the way, coincides with the nanoparticle recovery fraction. So therefore, we know that we've pushed, and we're now detecting the majority of DNA, of circulating, of cell-free DNA in the sample, in the fractions where the liposomes are coming out from. And not only that, we have different disease and different samples from different patients and different stages. In ovarian carcinoma, again, this is the studies I'm showing you. And we can see that we are consistently getting higher detection in terms of nanograms of cell-free DNA in the protein corona samples, which are shown here, compared to the healthy controls. And also, you see very clearly, with something that the oncologists knew, that, and they were expecting, they weren't surprised with it, that there would be more cell-free DNA in a diseased patient anyway. So that opened up in this paper that you can find here on our website. You can download all our papers free of charge, by the way, so please do if you're interested. That opened up this kind of spectrum. And this is a door for, I think, another laboratory, which is now being built in Manchester, not mine, because if you take this paradigm and you apply it, I do not think you can only get proteomic and genomic information. You can get transcriptomic, you can get lipidomic. So then we're talking about a nanoomic concept, which will allow you to perform with a single processing multi-omic analysis of the biofluid. And I think this is very important, and this is the way in which I think the biomarker field and the biomarker technology space is going to move, with combinations and correlations between different omics, as you can see here schematically. I will switch now to the graphene oxide technology. So this is a new nanoparticle in our initial two tables, okay? The younger toolbox that contained graphene. And the other very important thing is that this is not a spherical object anymore. This is a two-dimensional object. And it's not consisting of lipid, it's consisting of carbon. So here, just to put you on the kind of, there's a background slide of where does graphene fit. Graphene is a carbon nanostructure. You are all familiar with carbon nanostructures, by the way, as you can see here. So either as products of some kind of catalytic process, or whether you like chewing pencils when you were young, or whether you were given a diamond ring at some point in your life and your partner has not asked it back when the relationship ended. I'm speaking for myself. Anyway, and then we have the nanostructures here, which are the ones that the Swedish Academy of Sciences is feeling very fond of, clearly. So you have the structure of carbon atoms that is similar to a football, which is called a fullerene or a buckyball. It won the Nobel Prize of Chemistry in '96. Then you have the elongated hollow fiber shaped structure called carbon nanotube. The Swedes do not like long fiber shaped things, apparently, I don't know why. And then we have the graphene sheet, which obtained the Nobel Prize in Physics in 2010. Now, there is a reason, perhaps, why carbon nanotubes has not been awarded the Nobel Prize. And one of those may be that one could consider that a graphene sheet, so we're talking about carbon atoms only here. All of the structures I showed you are made of carbon, nothing else chemically. The only difference between all of those structures is the spatial arrangement of carbon, nothing else. So one could hypothesize, I guess, especially if you're a physicist, that the archetypal carbon nanostructure is a flat sheet. You just take carbon atoms, you lay them flat on a single plane, and you can generate all of these others. So perhaps this is an argument, but nevertheless, for me, as a hybrid between a chemical engineer and a biomedical scientist, it was fascinating to explore how those structures, flat structures, will behave in biological environments. And this was the reason why, as Xavier mentioned in the introduction of our history, is why I was recruited in Manchester, because Manchester got the Nobel Prize in graphene. This is one of the Nobel winners. And we sat down when we were putting the program together, and we were looking at what kind of information, what kind of data, what kind of knowledge is there available when you take a graphene sheet and you start interfacing it with biological entities, even a single mammalian or non-mammalian cell. And we found in this perspective article that there's all kinds of mechanisms that have been described. You know, there were descriptions, or one or two papers we're talking about. I'm not trying to give you the impression that there are hundreds of papers, by the way. Now there are more, but back then there were like 2-3 papers for each of these mechanisms. Some of them were talking about piercing plasma membranes. Some others were talking about integrating into the plasma membrane between the monolayers. Some others were talking about coating. You can even find papers about, for the microbiologists in the audience, of suffocating bacteria by wrapping up from graphene sheets. And then there's a lot of papers that are describing endocytotic mechanisms that are picking up the material, taking them in the phagolysosomal compartment, or others that are talking about translocation of the material into the cytoplasm. So you can find basically, you could find everything, anything. And there's a reason for that, and the reason comes back to the structure and the chemistry of these different structures, because you have different thicknesses, different surface functionalities, the material is extremely thick, and at the end of the day, you cannot control what people in one material, different laboratories are using. So when we set out the program, we said, okay, what are the unique properties that these materials offer that we can build something useful for biologists and clinicians? Because that's what I do. I don't care about sexy physics stuff, you know, they're good to have, but at the end of the day, somebody's got to do something useful with this. So there are a lot of different properties that are associated with the material. I'm giving you here this group of proteins that are, I colored green. Electrical mobility, fantastic. Flexibility, amazing. Heat dissipation, best in the world. Transparency, unbelievable. And the most important thing is that these properties are combined into this same single material. Now, you can find these properties from different individual nanomaterials, but this one has it all, in a way. We use these properties to design all kinds of devices. That was the heart of my presentation at the Institute of Neuroscience 3 weeks ago, so I will not discuss about this. If you want to know anything about this, we have to broadcast the talk, Xavier, I don't know. But what I will talk about, which I didn't touch upon really at all in this previous presentation, is this area here in orange, which is how does the material interact with different biological barriers and biological entities. Because that will determine not only the safety, toxicity, and public health implications from the interaction, but also it will give you the capacity to design transport systems of therapeutic agents. So let me illustrate this by giving you two examples, and I'm running short of time, so Xavier, please cut me. 5? Okay. No, not that. All right. Okay, so the first thing is that you need to be confident with your material. You cannot do this thing if you're not confident. So we spent like 5-6 years trying to optimize the material, what we call medical grade. What does that mean? Reproducibility, control of the lateral dimension. You see here atomic force microscopy images of these flat sheets. You want to make sure that the surface chemistry you perform is consistent, day in, day out, batch in, batch out. So whatever we do now, we have this checklist, which is like a materials data sheet. You know, when you order something for Merck or Sigma, which by the way sells the worst graphene oxide ever, so do not buy from Sigma. What I would advise you to do is look at how is it produced, what is the oxygen content, the purity, and most importantly for biological investigations that include immunological investigations, you need to make sure that it's endotoxin free. Because the worst thing can happen to you is if you have endotoxins introduced into your system, you don't know where, and something kind of activates, you don't know where the activation is coming from. So if you need these materials, we sell them at the Institut ICN2 free of charge. So we had very confident, very reproducible batches of these graphene oxide materials. We can now control the lateral dimension. We can have very tiny flakes. We can have small flakes. We can have large flakes. What do I mean large, small, ultra small? Small for us is about, as I showed you here, but I didn't spend time because Javier is forcing me to finish. It's in the order of 100 nanometers in lateral dimension. If you're talking ultra small, it's going to be below 100 nanometers. If it's large, it's around the micron size or above. Okay, that's this kind of scale we're talking about. Now you think of a typical mammalian cell is about 5 to 10 micron, just to give you a kind of dimensional check. Okay, so I brought two stories. I don't think he will allow me to give you two. I'll give you one. First story is to design a graphene transport system by intravenous injection. What do you do first? Well, first I want to know where does it go? What's the pharmacology of it? And the first thing we did is we tagged these sheets with radionuclides, and we performed pharmacokinetic, standard pharmacological pharmacokinetic studies, very traditional things. We checked our labeling efficiency, first of all. Check, it's over 99%. Then we moved on to labeling stability. We wanted to make sure that it's in the radionuclide in this chemical modification is stable in PBS and in serum, as you can see here. So we had a window of 24 hours that is very, very stable, so we were ready to go and inject intravenously. We took those sheets, we injected them intravenously, 50 micrograms in 200 microliters, a typical intravenous administration for those of you that are familiar with these experiments. And we performed Spec CT and gamma scintigraphy, which we have in our laboratory in Manchester, and we generated longitudinal pharmacokinetic profiles like this. So whatever you see shining is tagged graphene oxide. This is the zero time point. Zero time point means accounting for injection time, placing the animal in the camera, under anesthesia, blah, blah, more or less 20 minutes real time. So this is our time zero imaging. And then 4 hours after, and then 24 hours after. And you'll see very clearly that there are 2, maybe 3 sites of interest in this pharmacokinetic profile. Site of interest number 1 is this one here. What is this one here? Let me see the pharmacologist in the audience telling us. Bladder. So you see that we have a significant fraction that even within 20 minutes has come out of the animal into the bladder ready to be urinated. The animal has not urinated in 4 hours. That's why it's filling up, okay? And what happened 24 hours, the animal urinated and got excreted. So we knew that within the 24-hour period, we had a significant fraction of our injected dose that was excreting. That was interesting fact number one. Second tissue of interest is this guy here. What is here? Spleen, excellent. So this is the spleen. And then a third maybe tissue of interest is here, which is the liver. So we know very well. Now, if we do the same experiment with many more animals, and you do very traditional pharmaceutics now, where you what we call cut and count. You have many animals, you sacrifice, you take the tissues, you lyse them, and you measure with a gamma scintigraphy radio activity. You'll see very clearly, and this is very important and very useful for quantitative studies. You can see a confirmation of what we were observing here with the imaging. So spleen dose is accumulating as time goes by, and liver for some reason was higher and then it dropped. So we considered it that we were in the right direction. And more interestingly, if you look at the blood profile from blood detections, you see it's extremely fast. So those sheets do not spend a lot of time in the blood system, in the blood compartment. They distribute either through the glomerular filter or into the spleen very quickly. And this is a confirmation if you do the same experiment with animals where you collect feces and urine, which is an experiment that all of my students adore, then you can identify that you have a significant fraction of your injected dose excreted out in the urine and not in the feces. So the targets were identified physiologically, kidney and spleen. So kidney clearance. First of all, we wanted to map and see that our imaging is actually the imaging and not the detection of the label without that has detached from the nanomaterial. And we used Raman for this. So this is what the Raman is the signature that the graphene on its own is giving you. You don't need a label for a Raman pick, for a Raman signature. And this is the graphene oxide, the functionalized form of graphene oxide. This is what the Raman signature looks like. So then we started collecting urine, looking at the debris in the urine we could find on an optical microscope, as you can see here, and then going with a laser beam and trying to see if we do have graphene peaks. And as you can see here, we do. And once we start collecting this debris, we wanted to see if we have the structure of graphene out, and we have by electron microscopy confirmation that we do have intact the graphene coming out into the urine, which was very interesting. The next thing that we wanted to do is to understand how would these sheets cross the glomerular filter. And this is I have to admit, a fetish I have looking at the sections of the glomerular filter from past experiments and past life where we were trying to understand how fiber-shaped carbon nanotubes are crossing the glomerular filter. So just to navigate you a little bit on the electron micrographs I'm showing you here, this is the blood compartment and this is the urine compartment. And what you can find here is that you're having the glomerulus that is this barrier here. So when you're trying to identify these sheets, you need this type of microscopy that will give you high contrast of the sheets. So what you see here shining is the graphene oxides. I was trying to see if we could get sheets crossing as they cross, because they have to cross only in one axis, in one plane. If they try to cross flat out, they won't be able to cross, because the glomerular filter is not going to allow them to. So for some reason, there is a pharmacodynamic alignment of the sheets that would allow them to cross. And the whole experiment here is attempting to identify this. I think, personally, we failed with this experiment. We have not seen those sheets crossing. But it's an extremely interesting experiment. If anyone is a molecular dynamics expert or knows molecular dynamics experts crossing the glomerular filter, there are a few people like that. it would be very interesting to collaborate and see how these sheets are able to cross the glomerular filter, because we do see this consistently. We're talking 60–70% of injected dose coming out in the urine, as you saw, within the first few hours from intravenous injection. And there are many reasons we hypothesized in this paper why would this happen. So we couldn't identify them all, but we explained in the discussion part of this paper how could it be possible to have the sheets coming out into the urine from the blood compartment. There are many different types of options or possibilities, as you can see here. The sheet may roll, it can cross, it can crumble, etc., etc. So therefore, I'm going to close this by focusing on the second target, which is the spleen. And I think that also opens the possibility for a therapeutic development. If you look at the sections here, lung, liver, spleen, kidney, 0, 1 hour, and 24 hours after intravenous injection, it's very clear that we have material, the black material you see, in the spleen. So what we set out to do is, again, similar to what we did with the urine, identify by Raman and validate that the black stuff we see in the spleen is indeed graphene. And you see again this characteristic Raman signal, and then we go after spots in these black spots you see here in H&E, and we go under the microscope and focus the laser beam and get the Raman signal, and we can in that way validate that we do have graphene there. So we took this kind of principle one step forward, and we want to identify which particular cells in the spleen are responsible for collecting the material. And as you can see here, we stained anti-CD209 antibody, which is a macrophage stain, particularly spleen macrophage antibody. And we mapped this localization from sections to the Raman signal, to the Raman signatures. And you'll see here 1, 2, 3 corresponds to the Raman peak. So we knew that when we see those activated mature macrophages and we saw black matter it was indeed graphene oxide. And it's true that if you look at the schematics of the spleen histology, you see that the marginal zone macrophages are indeed the ones that will be theoretically collecting the material as it crashes and being absorbed by the spleen. So we cross-validated this by performing cell sorting with different markers, by, of course, breaking down the tissues and picking up different cells, and then co-registering the Raman signal. And we are now confident that we're getting about 50%, 40 to 50% of all of our macrophages, of our marginal zone macrophages, are Raman positive. So we know that these are the #1 cell population that is collecting the graphene oxide. And the next step is to do this longitudinally. How do you do this longitudinally? You say, okay, day 1, you got this graphene oxide landing in the macrophages. Day 30, what happens? You see again the stains of the macrophages, and then when you correlate it with the Raman, you see that you have, but the signal now starts getting distorted. So, and if you look at the Raman mapping in this bottom side, so this is the Raman signals, but as an intensity map. So the more yellow, red you have, the more Raman signatures you should be getting. But we start losing the Raman signature here. So why do we lose it? Do we lose it because the material breaks down? Do we lose it because the macrophages go take the material somewhere else? Or do we lose it because the material is starts moving to other compartments? And then we took it one step further and kept those animals for 9 months. And there you see you have a complete elimination of the Raman signal. You still have the marginal zone staining quite well, so the macrophages are still happy and healthy to pick up material, and they do stain well, but there's no material left. So then the question was, where is the material? And this is the closure of this study, whereby you see that by electron microscopy of the macrophages intracellularly in the tissue, you'll see that you're getting the material present even after 9 months, definitely within a month, but the material is no longer of the typical graphene structure. So it looks as if the material is partially degrading, partially transforming into this amorphous carbon mass. And I will leave you with this map, which is multiple studies after this, where we've identified now that the material is very comfortably internalized by all of these cells. Now, for the cell biologists in the audience, there will be many, you'll see that all of these cells are professional phagocytes. And then I think that what happens, similar to the spleen macrophages, professional phagocytes will maintain the material for a few months. They will start degrading them. They will be able to perform perfectly well on their own, but the material will still stay there. So then, because I've run out of time, you understand that we have the possibility here to design therapeutics and deliver therapeutics such as immunomodulatory agents, which is what we do and what was the last part of my presentation. And by delivering immunomodulatory agents into those cells, you have this window of about a month whereby you can present those cells and you can let those cells process immunomodulation agents through this kind of mechanisms. And this is the kind of direction at the therapeutic front that we are taking this technology. I will go fast. I'll skip the last part of the studies and go straight to my conclusion. I think I have persuaded you that nanotechnology is clinical reality. So I've talked to you about all these nanoparticles that are in the clinic that have a very rich clinical profile. I also think I persuaded you, or I hope I persuaded you, that you can use nanoparticles, even those that have a very rich clinical profile, and you can repurpose them, such as we did with the liposome story I told you, for the discovery or development of new tools. And I also think I communicated to you that, or I tried to communicate to you, that nanoscale technologies can help you interrogate and unravel mechanisms, such as the case of Alzheimer's, the Alzheimer's animal transgenic study that I showed you, but also in the design of new therapeutic modalities. And this is generally something I would like to emphasize. I think by understanding the fundamentals of pharmacology, physiology, and how these materials are interacting and being processed by the body, you are opening up possibilities for potential therapeutic applications. You could do it the other way around. You can stick a molecule, a therapeutic molecule, on the graphene oxide and start injecting it and see if it works. But for me, this is more like superficial science. I would always prefer if you go bottom up rather than going top down. But this is something I would like to leave you with as a potential kind of thought. We are open to biological and clinical users of our technologies, looking for these technologies to solve specific biological challenges you may have. So as Xavier mentioned, I'm very happy to discuss with you and meet with you. I am here in Barcelona 1 week per month minimum, so I'll be very happy to hear from you if you're interested. Of course, I wouldn't be able to give you this talk without all of these people. So we have 6 teams now in the nanomedicine lab. 5 are in Manchester, 1 here at ICN2. The Nanomics team is the one that is leading the technology on the biomarker discovery that I mentioned to you. The nanoinflammation team has been instrumental in the studies I showed you about how graphene oxide is processed by macrophages, microglia, blah, blah, blah. And the nanocell biology is a team that is looking at the interactions and intracellular mechanism trafficking of how nanoparticles are processed by different cell types, including the new thing that they're doing, which is organoids. And they started with lung organoids, in case you're interested. And the nanoneuro team is the team that I haven't really spoken about, because they're the ones that are using the devices and the neural interface devices we are building using graphene. And the nanotherapeutics team, I also showed you some of the data on the macrophage interaction. All of the materials, chemistry, and characterization of the material is now performed here at the campus at Bellaterra by the nanomedicine team at ICN2. Thank you very much for your attention.
Thanks, Kostas, for the talk. For some of us who knew the first talk, it has been absolutely new, incremental in knowledge, and I think that it opens a lot of stories. The session is now open to any questions, curiosities that you may have. I will bring you the micro.
Thank you very much for such interesting talk. My question is concerning the first approach of the liposomes as scavengers. What's the difference between the in vivo and ex vivo binding of the proteins in the liposomes?
That's a good question. The only difference we've seen with human samples is the fidelity of the. You can get a little bit more. It's richer. So I'll be very. I'm trying to abbreviate my response. The bottom line is that in vivo the environment is richer. The corona is richer. So we believe we get more information. We get more proteins. We get more smaller proteins, and it's consistently the mass spec data we are generating are consistently richer in content. For sure, you lose them, but that's why you want to be operating knowing the pharmacokinetic profile of your nanoparticle, because you know that, for example, from the graphene oxide, if I tried to do the same experiment with graphene oxides, it would have failed, because as you've seen from the blood circulation half-life of the graphene oxide, everything is already distributed in the body within, I don't know, 1 hour, and the typical in vivo extraction of the liposomes we did was 1 hour to begin with. So you can do it with 10 minutes. So we have a paper where we do 10 minutes, 30 minutes, and 1 hour. And when we were looking at the corona, if the time in which you let the liposome circulate longer, whether that makes a difference. But we knew again that we are in the bracket of pharmacokinetic profile where we have enough liposomes to perform the purification protocols.
What is the range of sizes of proteins that you can actually that can be absorbed by the graphene?
Oh, the liposomes, you mean?
Yeah,
or
I mean using the graphene or using the liposomes. What is the size of proteins that you can get? Even very small ones can be absorbed.
I mean, with the graphene, I'm not sure. I don't remember, to be honest, the range. With the liposomes, we get very small proteins, at very low molecular weight, up to albumins and immunoglobulins, and so I think but the question, you see me struggling here because I cannot distinguish between individual proteins and smaller proteins that are participating in a carrier complex. That's why you see me kind of hesitating here, because I cannot give you definitive data of single small molecular weight proteins that have directly adsorbed on the liposome or the graphene or the nanoparticle. Because how do you differentiate between those that are individually adsorbing and those that are part of a complex? I don't know. If anybody has any tool that can help us, it will be an interesting, but with the tools that we have, we cannot distinguish. We can say, yes, that by mass spec, the sequence is there, but that's an indirect, it's not a direct confirmation that you have small molecules that like to participate on the surface of the nanoparticles. Indirectly, you can hypothesize that because you are not masked by the highly abundant proteins, you can argue that the individual molecules find space which makes sense to absorb on their own, but I do not have an unequivocal evidence of this.
I can just ask. Hi, nice, very nice talk. Thank you very much.
Thank
This is our technologist. I was wondering whether internalization of graphene sheets by macrophages does promote or to promote reactivation, and whether these residents within macrophages alter macrophage activation profile once they are once they face
Yeah.
an activation challenge.
Yeah, thank you. The macrophages are activated. Well, it depends on the system you use, first of all.
It's quantitative, because I guess you don't know exactly how many graphene sheets do they internalize, no?
Correct. So the studies that I showed here with the spleen were not meant to examine the state of the macrophages. Just to clarify, for us the macrophages in these particular studies were the kind of dead end, if you wish, in the journey of the graphene oxide sheets. So then the question is, how does the macrophage that contains the material treat the material? Now, of course, you're asking me the flip side of this. How does the macrophage behave as it has the material inside? So you have to design completely different studies for this, I think. First of all, or you can interrogate this in situ macrophages differently. What we've done in the context of immunomodulation is we asked macrophages to pick up graphene oxide sheets and then challenge them challenge their phagocytotic performance, behavior, function, whatever you want to call it. And it looks as if you do not have a depletion. They can still phagocytose. Now, whether you can extend, but this is an ex vivo study. This is an in vitro, a cell culture study with bone marrow-derived macrophages. But whether I can extrapolate from these studies into the in vivo, I'm not sure. I don't know. I won't be able to answer the question. But the closest I can give you from the information we have is that macrophages in Petri dish, primary macrophages in Petri dish, maintain their phagocytotic capacity.
They are functionally very plastic macrophages, so they can be pro-inflammatory or anti-inflammatory. So regardless of their phagocytic capacity, do they change their functional profile upon activation by the fact of having internalized these graphene sheets? Have you tested or not?
We have tested, again, it depends on the type of macrophage. So we've done experiments with microglia, which you can consider. You can, or dendritic cells also are falling into this category. So every different cell type, yes, will have. Generally speaking, we know that the phagocytes are internalizing happily, in quotes, the material, and they can still generally function. That now, if you go into the very detail of how do you decipher function, that I'm not very confident, because I haven't done very detailed studies.
Any more questions? Maybe the last.
Perhaps you can hear me.
Oh, I can hear you very well.
Thank you. Thank you very much for your fascinating presentation.
Thank you.
I wonder is there any relationship between the liposome size and the sort of proteins that are absorbed on them? I mean, can you play with the size of the liposomes to get different results?
Yeah. We can play with the size of the liposomes, to answer your question.
Will you get different results or not?
So let me clarify. I want to make a declaration first. We have not done this experiment that you're describing. What we've done is we kept the size the same and we changed the surface. So we did cationic, anionic, polymer coated, blah, blah, 4 or 5 types, I can't remember. The protein content does not change dramatically depending on the surface properties of the liposomes. The total amount of protein changes. So cationic nanoparticles have more protein on it, but the molecularly is not a richer signature. Okay? Now, if you change the reason we have not done, or I'm a little bit reluctant to do this size-dependent experiment, at least in vivo, is because if we inject larger liposomes, I do not trust the pharmacokinetic profile of those structures. So I'm not confident that the pharmacokinetics will be the same in the interaction with the blood. But you can do the experiment ex vivo. We have not done the experiment, so I cannot answer your question directly as far as size is concerned. But the surface properties of the liposomes that we've changed did not alter the molecular signature in terms of quality, but the content, the amount of protein that you were getting was different. So cationic liposomes were getting more protein absorbed, anionic liposomes a little bit less, polymer coated liposomes a little bit less than the anionic. So that was a kind of spectrum, if you wish. Well, you got the microphone. You got the microphone, so you could. You got it in your hand, so.
But okay, I can. All right.
You can force the.
Okay, yeah, just briefly. I mean, you mentioned. I mean, really, thank you very much for the talk. Really, I also went to the other one, so I mean, I think I have. Probably you can give 2 or more talks, probably within the pandemic.
I haven't even told you about how we do the immunomodulation yet. So, and I think the lady at the back would be.
Yeah. So.
That's right.
So I was wondering about, you mentioned microglia, and I was wondering, I mean, are those experiments done like with microglial cultures? Because, I mean, the brain didn't seem to be a target. I'm assuming that because of the blood-brain barrier, probably. I mean, when you give the graphene, or how can you target, or do you know a way, or are you trying to figure a way to target the brain with a tail vein injection, or you have to go straight into the brain, or?
Yeah, I admit defeat by the blood-brain barrier many years ago. I admitted defeat. So I'll tell you why. Because I realized that the best liposome I could ever design, so in one of my postdocs, we had a brain delivery project, and I realized that I will, best case scenario, 3.5% of injected dose I'll get through. The magic transferring, translocating liposomes. So then, because of that experience, I kind of didn't want to address the blood-brain barrier. But this is a defeat of mine. Okay. So all of what we do is direct injections, either stereotactically or by convection mechanisms. So pumps, these pumps, I don't know if you're familiar with using neuro-oncology. So yeah, everything we do is directly administered, direct injections into the central nervous system.
Okay, and just to finish, I mean, it seems that the graphene sheets, I mean, they seem to be targeted to, I mean, microglial cells or macrophages.
Yeah, there is a...
Is there any other graphene-based nanoparticle that can target different cells, like neurons, for instance, or some other glial cells, or is it
generally speaking, these cells are, I mean, targeting is because the cells decide the targeting, not us, in a way. So it's like an opportunistic targeting, let me call it. So, but the answer is, even if I don't know if I showed you in the previous, I had the second part of the graphene story, had glioblastoma injections, so inside orthotopic glioblastoma, where you inject the graphene oxide into the tumor and into the brain. And you run the comparison, normal brain to glioblastoma. And you'll see there that even with cancer cells, you do not get a lot of internalization compared to the macrophage population that's in the glioblastoma mass. And in the brain, in the healthy brain, you see a very similar phenomenon. You see massive internalization by microglia. Very few material is entering into neurons. So I think all of the applications now we're developing with neurons is for the extracellular matrix and the plasma membrane.
Thank you.
Okay. Thank you very much.
Thank you.